Okay, we have DNA polymerase 1 and 3. Of course, the first function, 5' to 3' polymerization. There's no question about it. So what's the 1 for then? Cut Okazaki segment. Okay, what's the 3 for? Proofreading. That's a fidelity issue of the DNA. Okay? Then what is Okazaki fragment? Because at a lagging bend, you cannot do naturally 5' to 3' polymerization, so we need to synthesize those small segments. That's called Okazaki segments. Initiated with RNA polymerase. Okay? Okay, next couple of questions are a little bit difficult. And this is usually confuse people. Shown below is a nucleotide sequence of a fragment of double-stranded DNA from E. coli. The start site of transcription is indicated by the lowercase a. So where the start site is right here. First question, on the sequence above, show label where the promoter is located relative to the start site of the transcription. Then you basically need to know what is the sequence of the promoter. That is -10 TATAAT around the 16-18 base pair. Upstream, you have -35 sequence is TTGACA. So if I talk about that, did you figure it out? Yes, because we already explained. But if I use teaching, I just started, today is new, I read through the slides, can you understand it? I don't think so. That's the difference between when you write on the blackboard and you talk about on the slides. You see the difference? So, what will be the answer? Then you go back to see. Where are TATAT? Right here. Where are TTGACA? Is right here. So where are the promoter? You should be the, okay, you can, that's always trouble. Go this way, go this way, go this way. You have to go here. This whole thing is actually the promoter. Okay? This, you have a draw a circle on the top. That is RNA polymerase. So that's a promoter. Label the promoter. You can label this as -35 and then -10. We usually give you the 4 points. I'm not that picky. If you didn't label here, that's okay. Because I know you know this is -10, this is -35. But where is the answer? The answer is right this slide. Okay, so that slide is corresponding to that question. Name the enzymes that recognize the promoter sequences. This is easy, RNA polymerase. Name the protein subunits that allows the enzyme you identified in the question 40 to recognize and bound into the promoter sequences. What's that? Sigma factor. If you write the sigma 70, that's fine. Okay, that's what we talk about in the class. But best answer is sigma factor because there's a different type of the molecular weight of the sigma factor. Sigma 70 is responsible for the bacterial growing. Sigma 28 is for flagellar rotation. We have a different one. So this question actually is tested right here. Sigma 70, sigma 32, 54. And this question is actually for this one. Is that right? But this is kind of confusing you, so I usually draw on the blackboard, talk about alpha, alpha prime, beta, beta prime, omega. So that's the subunits is sigma factor. Right out of the MA transcription, be sure to label 5' to 3' ends. This is a question some people confusing. There is a people who is students start from beginning to the end to write it. Not really. What is the initial code? Remember, This is what we call it, coding strand. This is a template strand, is that right? The real mRNA sequence down here should be exactly the same as this coding strand. And exactly the same, the only difference is uracil replaced with T. But don't forget, start from transcription. There is an initial codon for it. What's that? ATG, which is AUG. So you only need to write something. This is. Okay? Is that easy? Yeah, that's what you should write. And the 5' to 3', the same thing. Because the top one 5' to 3', bottom one 3' to 5', then the transcription one is still 5' to 3'. Okay, so start from there. Not the whole thing. On the mRNA transcription, circle the second translated codon. Indicate which amino acid is coded by this codon. Well, second one is ATC. Is that right? Then it should be AUC. What's that? And if we need it, I'll give you the code. The one, I don't have to remember it. AUC. Isolysine. AUC is right here. Okay? This is a very good question. You should review. Because this question, you should put 2 stars there. This is a very good question to talk about the transcription. And every single piece of information is right there. That's why I want you to practice. Because we talk about the promoter sequence, the RNA polymerase, and we talk about sigma factor, we talk about how to write the mRNA transcription coding. That is starting to find AUG. And we also talk about the codon, the 3 letters of codon. So, it's a very good question. You should mark it to study. Gene expression, translation. Shown below is a partial sequence of an mRNA, of an RNA molecule. Could this RNA transcript be an mRNA? Why or why not? And I think you don't know, so I'll write a clue. What's the significance of the underlying sequence? The answer is yes. Why? What is AGG? AGG means? That's Shai-Dagano sequence. Yeah, of course you can do it. Would the deletion of the base marked G1 have any consequences? So, what do you need to find? AUG is the start codon, started to do transcription. If this G is taken out, what happened? The reading frame will be shifted, is that right? So the transcription could be stopped. Then the last question, what would be the consequence of deleting a lower case bolded right here on the sequence above? Well, that's a debate question. It could be have impacts, it could be no impacts. It depends on the reading frame. Because if this A in a certain position of a codon, then it doesn't matter sometimes. Because we call it a wobbling impacts. Okay, that's a debate question. Okay, very good. So these are we studied. You don't have to worry about basically 44 and 45 because this is just to let you understanding a little bit because we didn't talk about the reading frame shifting. Okay, but you do need a study, which is 43, which is from 39 to 43, because these questions are really nice to let you understanding the transition between transcription and translation, what the story it is. Okay, the other 2 questions are relatively difficult. It's just let you think a little bit. Okay, so we finished this one. Then we have a last set of the question. So let's go over it. And the key will be put on the eCampus tonight. And another thing is that Carly Long will offer additional review section tomorrow afternoon, 4 o'clock in the lab room. If some of you still want to help, she will help because she is a biochemistry major student. And she can go over these with you guys if you're still confusing. Okay? That's the last minute review. That's good sometimes. So let's look at all these questions. Coenzyme. Small protein function transfer specific type. The answer is D. Some small molecules derived from vitamins. Okay. Sulfur drug, we already mentioned, inhibit synthesis vitamin by, we already talked about that. Okay, that's C. Same question. Transfer electron dehydrogenase is called, this is what we already talked about. What is this called? Fermentation. Okay, this one. The purple sulfur and the green sulfur bacteria obtain energy from growth and anaerobic respiration. Okay, then this is something. By oxidizing organic molecules, particular acetone, no. From photosynthesis, no. By oxidizing of NH2 from the reducing of, which one? This should be E. Is that right? The reducing sulfate go to hydrogen sulfide. That should be E. The same thing like here. Same thing like here. Okay. Which of the following is best definition of fermentation? Oh, oxygen, oxidation, carbohydrate with organic molecules serving as final electron acceptor. Okay, this one. At the bottom of the swamp, you may find a black deposit produced by the metabolism of disulfur vibrio. Look at this word. What is a vibrio? Is morphology disulfur, which tells you something. This bacteria using what as its terminal electron acceptor? The answer should be H2S. But sometimes SO4 could be, but is not, is limited. So the best answer is H2S. Okay? This one, if you really wanted to see, this is used to be, we had a question which is a little bit difficult. Let you want to sort about, let me find where should be this answer. This is somehow here. Disulfur is right here. Disulfur vibrio. Electron acceptor is this, reduced product is H2S. Okay? So, terminal electron acceptor is sulfate. But it's okay if you do H2S. I know you understand. At least you already know it's sulfite. Okay, so the year what I graded is that if you do SO4 and H2S, both are okay, but the best answer is SO4. Because I know you understand it's sulfite. I'm not that picky for the reality, the detail of it. Okay, because we didn't emphasize too much. You don't have to do that one. That's too complicated. This is a fermentation. I can tell you this is CH4 and anaerobic respiration. This is too complicated. The mud, you didn't see that anymore because this is what happened all the time. They're using the mud. The CH4 is an energy resource. They saw it's a clean energy by all the time. Where it come from? Come from the human animal poop samples. We don't do that anymore. So I just skip that question because it's out of dated. Not much meaning to do it. Which of the following reactions take place outside of the bacterial cell? Then you tell me which one. Remove of electron and proton from organic chemicals by dehydrogenase. This should be happen outside of the cell wall because this generates proton multiforce. Which is the best definition? Anaerobic respiration. Inorganic molecule as a final electron acceptor. So all these questions, when you see the best definition, you could combine them to study. That's all the same thing right there. Conversion is phosphoenolpyruvate to pyruvic acids. Involves conversion ADP to ATP, involves substrate level phosphorylation. So what's the answer? B and C, yes. And the time when I have this question is difficult because I don't talk about those details. I started to draw everything myself is like 3 years ago, right before 4 years ago, right before the pandemic. Before I talk, I just said, oh, here is the ATP, here is the ATP used, here something, here something. I just teach that like 15 minutes. But I get the review feedback from students. They say, don't take it for granted we know biochemistry. That's why I started to draw all the details. Okay, that's what the feedback from the students. So by the time, this is difficult because you need to know where is the phosphoenolpyruvate. And this is a big chemical. Remember, group translocation is used that chemical. So that's a good question. How does fermentation of glucose differ from oxidation of glucose? Yes, that's the first one. Fermentation produces less glucose. Yeah, that's B is good. But when it oxidative respiration, that's ABC, I would say. Okay. How are anaerobic reactions similar? Yeah, both use inorganic molecule as a terminal electron acceptor. The primary purpose of a catabolism. Okay, what is catabolism? Generate ATP. That's the purpose for catabolism. Then the anabolism will use ATP. Is that right? So the primary purpose, generate ATP. And the year, something people say this, that's okay. If I see you pick D, I still give you the points, but the best answer is generation of ATP. Okay. What is this one? Involved in oxidation H2S2S. Well, this is tough. Then this one you have to go back. I will tell you it's a bigota. That's the answer. Okay. This will not test you. Just to give you a concept, there are some sulfur bacteria there. 1 turn of Krebs cycle generate what? This you should know. Of course, E is gone. There is no ADH there. Of course, A is gone, because there is no what? There's no FADH there. So what is it going to be? Going to be right here. This is when we talk about the 1 turn of Krebs cycle. The question here did not include the transition step. So don't be confusing. But I will write clearly at the top of the question, related to or not to the transition step. You don't have to do that one. Chlorophyll, we did the mention. Photo, that's water. You don't have to do it. All these, you don't have to do it. Yeah, you don't have to do these. So good. So, what are the key to study every single question of the practice exam? Watch my 2 videos for the extra exam. Don't forget about these. And attend the special review section if you need it. Okay?